Hello, biology students! Now we're going to talk about how DNA likes to replicate or copy itself, which is super important for how cells divide, and we have identical cells throughout our body with the same DNA, even though some of those cells do tons of different functions. They all have the same DNA. Whoa! Before we jump into this too deeply, we do have to have some basic history about how DNA was discovered. First, there was a really young woman named Rosalind Franklin, and she worked with a man named Maurice Wilkins. They were the first to study the double helix shape, and they studied it using this photo that's very famous called Photo 51. This photo has an X-looking shape across it, which suggested that DNA was a double helix, not a single or a triple helix, because guess what? People didn't know before then. Whoa! But keep in mind, this was a long time ago, and Rosalind Franklin, well, was a woman. So unfortunately, even though we give her credit now, the Nobel Prize went to James Watson and Francis Crick, because Maurice Wilkins was talking to them, and they ended up stealing some of Rosalind Franklin's images, such as that previous photo, and they used it to actually get the Nobel Prize for discovering the shape of the double helix. So poor Rosalind Franklin didn't get credit. She wasn't very happy or nice about it, and it's really depressing because also she was doing so many x-rays that she ends up getting too much x-rays on her body, and it causes her to get early cancer, and she passes away. She never ends up living to know that she now has some credit given to her. Well, our major focus is based on the fact that DNA is a double helix. It's able to make copies of itself. And DNA replication is referring to how we can go from having one double helix to making two identical double helices, which means two double helixes. How does this happen, and when does this happen? Well, this happens before a cell can divide. We're going to learn later in the year, we've already maybe mentioned that cell division is called mitosis. But before the cell can actually divide through this process called mitosis, DNA has to have identical copies of itself in the cell. And the time period when DNA does this copying is called interphase, the time in between cell divisions, in between. So in between when the cell divides, the DNA makes a copy of itself. This is a really long period of time because copying is really important, and we need to make sure that the copies are exactly identical. Now let's learn how the copying happens. Well, it happens because DNA is referred to as a template. What's a template? A template means it's a pattern that can make copies off of itself. So sometimes we see template referred to as the same thing as a negative for photos, but guess what? You guys aren't so used to having photo negatives now that we have so many digital photos. But back in the day, this type of thing could be taken to a store and lots of copies could be made of that photo. Nowadays, template sometimes is used to talk about a file on Google Docs that you guys can make a copy of if the Google Doc is view only. A view only Google Doc is a template if you had to make copies off of it. Interesting. But the important thing is DNA is able to make lots of copies of itself because it is referred to as a template. One strand of DNA can make two identical DNA copies, which is really cool. And this can happen over and over and over again, which is really neat. What are the steps of this process? Well, there's three major steps to how DNA duplicates or makes a copy or replicates itself. The first step is that DNA has to untwist and unzip. Remember that DNA is considered a twisted double helix ladder. Well, we need to untwist it so that it looks like a regular ladder shape. So that way we do that is it needs to happen fast. And what type of things do things fast in the cell? For chemical reactions. Oh, enzymes. Enzymes do things fast in the cell. Well, the specific enzyme that does this untwisting and unzipping is called helicase. Sounds like the word helix because it untwists the helix. Helicase does this. It breaks the hydrogen bonds between the base pairs, and it pretty much, after it makes it into a regular ladder shape, it treats it like a zipper and it unzips the DNA. So first we unravel it, and then it opens it up. Super cool. So that's the job of helicase. Helicase does this super fast because it's an enzyme. The next step is that it adds nitrogen bases. The nitrogen bases get added in a very specific way, where C always goes with G, and A always goes with T. So we could consider this base pairing rules. So nucleotides that are floating around called free nucleotides, C's, G's, T's, and A's, get added to the open template strand. Let's look at that again. We would consider this the template strand, and now those free nucleotides are being added to that open strand using base pairing rules. Because this happens fast, another enzyme is involved called DNA polymerase. Why is it called polymerase? Because it's making a polymer. ACE usually refers to the fact that we're referring to an enzyme, and it uses DNA base pairing rules. That's why we call it DNA polymerase. Last step, the end result is we have two identical DNA strands. All right? And then, why is it that one half has old and the other half is new? And that's the same on the other side. See, we have half blue and half yellow. Well, that was because DNA was a template. The blue part was the original. We call this a semi-conservative process because half of the original still is around. The template is still conserved or existing, somewhat conserved. So the way we define semi-conservative is each new strand is made up of one new DNA piece and one old parent DNA piece. Somewhat new, somewhat old, semi-conservative. Wonderful job, guys. We'll practice all these terms and even practice doing some DNA replication in class. See you then.